Welcome students to my channel Biology by Dr. N.S. Jain. Today we are going to study a very important topic related to the cell biology, and that is the type of the cells, which is equally important for the class 11th. It is very important for all the medical entrance examinations. Types of cells. Here there are three types of cells written: prokaryotic cell, mesokaryotic cell and eukaryotic cell. Very first type of the cell is the prokaryotic cell. Pro means primitive and karyotic means nucleus. This prokaryotic cell is the feature of the primitive organisms. Here the nucleus is not well organized, means the nucleus is not bounded by a well defined nuclear membrane, and such a nucleus is called as nucleoid or genophore or primitive nucleus. I am repeating once again, a nucleus is not well organized here, it is not bounded by a nuclear membrane, and such nucleus is called as nucleoid or genophore or the primitive nucleus. One more feature of the prokaryotic cell is that here the DNA is naked, means here the DNA is not associated with the histone protein. DNA is naked and the histone proteins are absent here. There are three examples of the prokaryotic cell: the bacteria, the blue-green algae and the mycoplasma. Here the blue-green algae means cyanobacteria. The second important type of the cell is the eukaryotic cell. Eu means true and karyotic means nucleus, means a eukaryotic cell has the true nucleus, while in the case of the prokaryotic cell, the pro means primitive and karyotic means nucleus. Here the nucleus was primitive, here the nucleus is true. Now the question arises, what do you mean by a true nucleus? True nucleus means the nucleus is well organized, means it is bounded by a well defined nuclear membrane. Not only this, here the DNA is associated with the histone protein. Like the prokaryotic cell, here the DNA was naked and the histone proteins are absent. You can see that here the DNA is wrapped around by the histone protein. You can see here I have drawn a diagram. This is what the histone protein. This is the histone protein and this is the DNA which is wrapped around the histone proteins. Now all the higher plants and the animal cell are the examples of the eukaryotic cell. The next cell is known as the mesokaryotic cell, and the mesokaryotic cell is having the characteristics of both the cells. It has some characteristics of the prokaryotic cell and it has some characteristic of the eukaryotic cell, means we can say that these cells are having intermediate organization. They are having prokaryote as well as eukaryote characters. Now the question arises, which is the eukaryotic character and which is the prokaryotic character found in a mesokaryotic cell? So here you can see that the nucleus of the mesokaryotic cell is bounded by a well-defined nuclear membrane, which is a eukaryotic character which you have discussed just now. The second is the histone protein is absent. We have discussed just now that histone proteins were absent in the prokaryotic cell. So histone protein is absent. So they are having eukaryotic character, they are also having prokaryotic character, means altogether we can say that they are having some characters of eukaryotic cell, some characters of prokaryotic cell. Now the best example of the mesokaryotic cell is the class of an algae known as the Dinophyceae. Dinophyceae members have mesokaryotic cell organization. Now just now we have discussed about the prokaryotic cell and the mesokaryotic cell and the eukaryotic cell, but here we have discussed only 2 or 3 points of the prokaryotic cell and the eukaryotic cell, but now we have to discuss few more points which are very important to discuss for the prokaryotic cell and the eukaryotic cell. So let's have a discussion on the differences between the prokaryotic cell and the eukaryotic cell. Now there are many differences of prokaryote and eukaryote. We will be differentiating by table. See here, I have made a table here. Here I have written the characters. Here prokaryotic cell, and here eukaryotic cell. Now see, point number 1, cell size. That what is the cell size? So always remember that a prokaryotic cell is small in size than a eukaryotic cell. Here they are small, they are having the cell size of 0.5 micron to 5 microns, while a eukaryotic cell is large and it is having the size of 5 micron to 20 micron, and these cells are primitive, while the eukaryotic cell are advanced. Second point, cell wall. In prokaryotic cell, the cell wall is made up of murin or peptidoglycan, while here in the case of the eukaryotic cell, the cell wall is made of, in plants, made of cellulose. In animals, the cell wall is absent, and in the case of the fungi, the cell wall is made of chitin. Got it? Here the cell wall is made up of murin or peptidoglycans. Also we can say that some amino acids and carbohydrates are also present in the cell wall. Here in the case of the plant cell, it is made up of cellulose. In animals, the cell wall is absent. In fungi, it is made up of chitin. Now coming to the third point, cell membrane thickness. That what is the thickness of the cell membrane? Here the thickness is 40 angstrom to 50 angstrom, while here the thickness of the cell membrane is 75 angstrom to 100 angstrom. Fourth point, mesosome. Here the mesosomes are present. Now see here, one thing to be noted here is that in prokaryotic cells, mitochondria are absent. Now they are not having any energy source. Then the question arises that how the cell will get the energy. So the cell will get the energy from mesosomes. Instead of the mitochondria, a prokaryotic cell is having mesosomes. Now the question arises how the mesosomes are formed. So see here that this is the bacterial cell. Say for this is the bacterial cell and this is the cell membrane. So what happens that this cell membrane fold inward. This cell membrane fold inward and it forms mesosome. So this is what? Mesosome. So mesosomes are nothing, they are formed by the infoldings of the cell membrane and they work just like the mitochondria. So here the mesosomes are present, but here the mesosome is absent. Why? Because mitochondria are present. So there is no use of the mesosome, so mitochondria are present here. Now, respiratory enzymes. Those enzymes which are used in respiration are called as respiratory enzymes. So the question arises that where the respiratory enzymes are present in a prokaryotic cell. So in prokaryotic cell, the respiratory enzymes are present in mesosomes. Respiratory enzymes are present in mesosomes. Have respiratory enzymes. While here, as mitochondria is present, and we know that in the matrix of the mitochondria, Krebs cycle continue. In the oxysomes of the mitochondria, electron transport system is present. We can say that here the respiratory enzymes are present in mitochondria. तो respiratory enzymes present in mitochondria. Got it? The next point is that membrane bound cell organelles, what are membrane bound cell organelles? You have studied certain cell organelles, mitochondria, Golgi body, endoplasmic reticulum. All these membrane bound organelles are absent here. In prokaryotic cell, membrane bound organelles are totally absent. So the membrane bound organelles are absent. I want to say, for example, mitochondria, Golgi body, ER, etc. are absent here, but in a eukaryotic cell, all membrane bound organelles are present. Next is the nucleus. Well organized nucleus absent. Here the nucleus is not well organized. Nuclear membrane absent, while here well defined nucleus bounded by nuclear membrane present. Once again you can see the difference. Cell size, here the size is small, here the size is large. This cell is primitive, this cell is advanced. Cell wall made of murine, peptidoglycan, amino acids or carbohydrates. Here the plant cell is made of cellulose. In animal cells the cell wall is absent. In fungi the cell wall is made up of chitin. The third point, cell membrane thickness. The cell membrane thickness is 40 to 50 angstrom. Here the cell membrane thickness is 75 to 100 angstrom. Mesosomes. In place of mitochondria, prokaryotic cell have mesosomes, which are the enfoldings of the plasma membrane. Here mitochondria is present but mesosome is absent. Respiratory enzymes are present in mesosomes. Here the respiratory enzymes are present in mitochondria. Membrane bound organelles, all the membrane bound organelles are absent. Here the membrane bound organelles are present. Nucleus, very well defined nucleus bounded by nuclear membrane is absent here. Well defined nucleus bounded by nuclear membrane is present here. Few more differences. Let's talk about the nucleolus. What about the nucleolus? That whether it is present in the nucleus or not. So the 8th point is nucleolus. As the nucleus is not well bounded by the nuclear membrane, here the nucleus is absent. Nucleolus is absent. Here the nucleolus is present. Now the ribosomes. Ribosomes are also cell organelles, but they are membraneless. So they might be found in prokaryotes also and also in the eukaryotic cell. Here 70S type of ribosomes present. Here 80S type of ribosomes present. Now histone proteins. These are basic proteins which are absent here while they are present here. They are of 5 types: H1, H2A, H2B, H3 and H4. Now the DNA, as the nuclear membrane is absent, so it is scattered in cytoplasm and less in quantity. Here the nucleus is well defined, so DNA is present inside nucleus and more in quantity. Next point, if we talk about the genetic recombination and sexual reproduction, so here in prokaryotes only genetic recombination present, true sexual reproduction absent. True sexual reproduction absent, while here true sexual reproduction present. Next is the cilia and flagella. If we see the internal organization of the cilia and the flagella, so they are made up of microtubules. So here 0+1 arrangement of microtubules present, while here 9+2 arrangement of microtubules present. Once again, nucleus absent, here present. Ribosomes, which is a membrane bound, which is membrane less cell organelle, 70S type of ribosomes are present. Here 80S type of ribosomes are present. Histone proteins, which is the basic protein, is absent here. Here all the 5 types of the histone proteins present. DNA is scattered in cytoplasm as the nuclear membrane is absent and it is less in quantity. Here the nucleus is well defined, so DNA is present inside nucleus and more in quantity. If we see about the genetic recombination and the sexual reproduction, here only genetic recombination present, true sexual reproduction absent. Here true sexual reproduction present. Flagella and cilia, we know that these flagella and cilia are composed of microtubules. Here the arrangement of the microtubules inside the cilia and the flagella is 0+1 arrangement. Here it is 9+2 arrangement. Now, type of division. Here amitosis type of division present. Here mitosis and meiosis division present. Here amitosis, here mitosis. Next one, spindle fibers. Now spindle fibers are the fibers which take the chromosome towards the opposite poles, but here no spindle fiber formation, but here spindle fiber formation occur. If we talk about the vacuole, so vacuole is absent here. while the vacuoles are present. In a plant cell, big vacuoles are present, while in the case of the animal cell, small vacuoles are present. Nitrogen fixation. This power is the feature of the prokaryotic cell. So nitrogen fixation is present here, while in the case of the eukaryotic cell, the nitrogen fixation power is absent. Examples of the prokaryotic cell and the eukaryotic cell. Bacteria, as we have discussed, bacteria, blue-green algae and mycoplasma are few examples of prokaryotic cell, while eukaryotic cell, all higher plants and animal cell are the examples of eukaryotic cells. One more thing we will be liking to discuss about the linkage groups. That linkage groups, how many linkage groups are present in prokaryotic cell? So in a prokaryotic cell, the number of linkage group is only 1. Now why 1? Because they have single chromosome. They are not having the multiple chromosomes. They are having single chromosomes. So the number of linkage group is also 1 only. While in the eukaryotic cell, the number of linkage group is equal to haploid number of chromosomes. Number of linkage group is equal to haploid number of chromosomes. Means, say for if the human beings are having the 2n number of chromosomes 46, so the half number is 23, so linkage group of the human beings will be 23. If the maize is having the 2n number of chromosome 20, so the linkage group will be 10. And at the last, if we talk about the chromosomes, so here the chromosome is single chromosome. They are not having multiple chromosomes, they are having single chromosomes. And here in the case of the eukaryotic cell, multiple chromosomes present. There is one more difference in between the prokaryotic cell and the eukaryotic cell, and that is exocytosis and endocytosis. Exit of substances from the cell means exocytosis, and entry of the substances in the cell is called as endocytosis. But here the exocytosis and the endocytosis are absent, and the eukaryotic cell have both exocytosis and endocytosis. So these two phenomena are present here. So these were the differences between the prokaryotic cell and the eukaryotic cell. If you have liked this video, please subscribe my channel, like and share this video. And also students, if you are facing any problem regarding this topic or any other topic of the biology, you can comment in comment section. I will be giving you the answers of your question as soon as possible. Thank you. We will be coming soon with new videos with new topics. Thank you.